Now we're going to be going over Pacific Biosciences, which is another method, PacBio, and it is called as SMRT sequencing, which is single molecule real-time sequencing. And we'll discuss in depth why all these points have been listed. We'll just be going over the main points here, and then I'm going to go to the in-depth explanation of each point. Illumina amplifies a single molecule multiple times to build a cluster. Illumina we discussed in the last video. This results in a multiple fold signal increase as compared to a single molecule, resulting in accurately capturing the base. So PacBio uses a single large molecule as a template. Sequencing by synthesis in each template is recorded as a real-time movie. Hence, it is called single molecule real-time sequencing (SMRT). Moving on now, basically now I'm discussing Pacific Biosciences SMRT sequencing. So the core principle is that it is a single molecule real-time SMRT sequencing. First, let us see the difference, the fundamental difference between Illumina versus PacBio. So what Illumina used to do is basically fragments are amplified into clusters, bridge amplification. Then the signal comes from many copies, averaged per cycle. Then the extremely accurate per base, but limited read length. In PacBio, we have no amplification, right? No bridge amplification. Instead, a single DNA molecule is sequenced in real time as it is copied by a polymerase. Each base incorporation event produces a fluorescent pulse that is recorded live. Why real time? The sequencing reaction is observed continuously as the polymerase adds nucleotides. Each incorporation produces a measurable pulse of light. So the machine records a movie of the polymerase in action, base by base. Template is PacBio typically uses long intact DNA molecules, often greater than 10 kilo base pairs. So it can generate very long reads in as compared to Illumina. So why it is real time? Because sequencing is basically observed continuously as the polymerase adds the nucleotides, right? And each incorporation produces a measurable pulse of light. So machine records the movie of the polymerase. Now, how does it record a movie? That we'll discuss using the ZMW technique. That is the zero mode waveguide, the heart of SMRT technology. What is a ZMW? So a ZMW is a nano photonic well, which is a tiny chamber close to approximately 100 nanometers wide. That is very small, microscopic level, right? 100 nanometers wide, etched into a metal film on a chip, right? A tiny chamber which has been etched into a metal film on a chip. Light penetration into this well is so shallow that only the very bottom, where the DNA polymerase sits, is eliminated. So set up inside a ZMW, which is the full form is zero mode waveguide. So a single DNA polymerase is immobilized at the bottom, right? So we have something like a well shape, right? A single DNA polymerase is basically immobilized at the bottom. A single DNA template molecule is threaded into polymerase like this. Fluorescently labeled nucleotides, each base tagged with a different color, float freely in the solution that we have, right? We have a solution. How it works: when the polymerase incorporates a nucleotide, the fluorescent tag emits a signal. Once incorporated, the tag is cleaved and diffuses away. So only the correct incorporated nucleotide gives a brief pulse. Each pulse is one base call. Now we have parallelization. So thousands of ZMWs on one chip work in parallel. So thousands of single molecules are sequenced simultaneously, analogous to how thousands of Illumina clusters are sequenced in parallel, but here it's one molecule per ZMW. Output: each ZMW generates a movie of light pulses which are interpreted into the base sequences in real time. Now, key highlights. So the comparison slide to Illumina, basically Illumina clusters versus PacBio ZMW spots. So in Illumina, one fluorescent spot is one cluster of many molecules. One fluorescent spot, one cluster of many molecules. But in PacBio, one fluorescent spot is only one ZMW, and it refers to only one DNA molecule, right? So that is the difference. Here we have multiple molecules within a cluster, which is denoted by one, which is shown by one fluorescent spot. And in PacBio, one fluorescent spot corresponds to one ZMW, which means we have one DNA molecule only, right? So that is the main difference. And the signal strength in Illumina, we have a strong average signal with low error, but requires PCR amplification. In PacBio, we have weaker single molecule signal, higher raw error, but avoids PCR biases and preserves very long fragments, and there is no amplification. Now moving on, advantages of PacBio SMRT sequencing. So it has extremely long reads, right? So as compared to the Illumina that we discussed, Illumina used to have like close to 150 to 300 base pairs as the read length. But in PacBio, we have typical read length of close to 10,000 to 30,000 base pairs. That is much more than the Illumina base pair read length, right? Illumina read length. So with some reads exceeding 50 kilobytes as well. So that is extremely long read. It what extremely long read does is that it enables assembly of complex genomes, resolution of the repeats, and direct haplotype phasing. In microbiome or metagenomics, it allows full-length 16S rRNA genes to be sequenced in a single read. That is basically a single read of this full-length 16S rRNA genes is around 1500 base pairs. So easily it can accommodate that, providing better taxonomic resolution than Illumina short amplicons. Right. Now, next is high throughput. That is another advantage. Modern SQL2 systems, one SMRT cell can guarantee tens of gigabases of sequence, suitable for large-scale projects, though historically lower throughput than Illumina. Then also it has no amplification bias since DNA is sequenced directly without the PCR. GC-rich or AT-rich regions are less biased. It is important for accurate genome-wide studies, for example, identifying structural variants. Also, it gives kinetic information, which is unique. The polymerase kinetics, example, pause duration, can be recorded, giving epigenetic data like DNA methylation detection without extra library prep. Now, going over the disadvantages of PacBio. So it has higher error rate than Illumina. That is because it has raw reads. Historically, 13 to 15% per base error, mainly insertions or deletions. Modern hi-fi CCS circular consensus sequencing, multiple passes of polymerase over the same circularized template yield consensus reads of 10 to 25 kilo base pairs with greater than 99.9 accuracy. This solves the error issue, but at the cost of requiring smaller templates for multiple passes. Then higher cost also is there. Instrument and consumable costs per base are higher than Illumina, though cost per GB has dropped significantly with SQL 2e. Best suited when long reads are essential, for example, resolving structural variants, metagenome assembly, full length rRNA as well. Throughput versus Illumina, still generally lower throughput than Illumina NovaSec for massive short read datasets. For very large population studies, for example, greater than 1,000 samples, shallowly sequenced, Illumina is cheaper and more scalable. Now, finally discussing where PacBio shines, some examples. Microbiome full-length 16S sequencing. It avoids the limited resolution of V3, V4 amplicons sequenced on Illumina. Then PacBio can differentiate closely related species, for example, E. coli versus Shigella, that are indistinguishable with partial 16S. Then de novo genome assembly, closing microbial genomes, right? Now, after that, epigenetics, detection of 6 milliamperes and 5 millicapacitance methylation in bacterial genomes directly from kinetic signals. In short, PacBio SMRT uses ZMW, uses zero mode waveguide nanophotonic chambers to record polymerase activity in real time, yielding extremely long reads, up to tens of kilobyte. Its strength are long contiguity, no PCR bias, and unique epigenetic detection. Weaknesses are higher cost and higher error rate, though modern hi-fi reads combine the length advantage with Illumina-like accuracy. Compared to Illumina, PacBio sacrifices throughput or cost efficiency for a depth of information. That is, it gives long reads plus epigenetics. So that's it.